The overall goal of this procedure is to isolate epithelial cells from the rat epididymis and to measure the electrical properties on these primarily dissociated cells using whole cell patch clamp method.
This method can help answer key questions in the reproductive biology field, such as the function of epididymis, which is important for sperm maturation and inheritance of paternal traits. This technique allows the user to characterize the electrical properties of isolated epididymal epithelial cells so that unknown physiological function of these cells can then be elucidated. Demonstrating the procedure will be Zhang, a grad student from my laboratory.
To begin the procedure, disinfect the dissection tools by immersion in 70% ethanol. Then, let them air dry. Next, prepare the full IMDM. Afterward, create 50 ml aliquots in aseptic conditions, seal with parafilm, and store at 4 degrees Celsius. Following that, prepare a collagenase enzyme digestion solution by dissolving collagenase type 1 and collagenase type 2 in rPMI, resulting in 1 mg per milliliter of each collagenase in the solution. Filter the solution through a 0.22 micrometer membrane. Then, mark it as collagenase solution and keep the solution at room temperature until use. Next, adjust the volume of the enzyme solution based on the weight of the enzyme.